Today we will discuss about one of the recent published paper about the type 2 diabetes possible treatment, and we will discuss how they have did this uh, type 2 diabetes possible treatment in one of the patients. So what they did is they take the peripheral mononuclear cells, peripheral blood mononuclear cells from the patients, and then convert them into this humanized pluripotent stem cells. And after that, they differentiate themselves into this uh, neural stem cells, and after this neural stem cells they further differentiate themselves into these islet cells, and the islet cells has the property to secrete hormones, and that in the pancreatic islet cells can produce this glucagon and insulins. And what they did is they transfer these islet cells into the patients, and before doing this step they also validated their islet cells in the mouse models for the safety concerns, and they are able to treat one patients uh, who is infected with type 2 diabetes. So some of the data about the mice are presented here. So for example, they take the mice and induce these STG inductions, thereby they make the mice that has the diabetes. And uh, as a control model, they did not introduce islet cells in the one of some of those diabetic mice, whereas some of these diabetic mice has been introduced, or kidney capsule transplantation occurs of the some of the diabetic mouse with islet cells. And what they have find out is that the control mice are died because of hyperglycemia and death. That leads to this death. Hyperglycemia is the conditions where high blood sugar level leads to the death of the mice. Whereas in the case of the mice that is infected with islet, they can see the reversal of hyperglycemia, and the mouse is able to survive. And when they check their glucose level in those mice, they can see that uh, the control mice has the sustained glucose level, whereas for this mice that are transplanted with the islet, they can see the decrease in the glucose level till this nephrectomy. Nephrectomy is a surgery where they will remove the kidney, and since these islet cells are introduced in the kidney, so once the kidney is removed, they can see the again the increase in the glucose level. And they also checked one of the signature proteins that are produced whenever the whenever the insulins are secreted, that is human C peptide, and they can see that human C peptides levels are high at the 19 days, and also the human C peptides are high at the 19 days when the glucose is introduced. This measurement is 30 minutes after the glucose injection, whereas the fasting rate, the human peptides are C peptides are low, and after 180 days they can see there is also again the increase in these human C peptides when the mice is injected with, when the mice has this glucose. This data is about 30 minutes after the mice has taken some meals that has this glucose. They can see the insulin productions, which with the help of the signature protein, that is human C peptide. So after the successful results or the expected results they get from this SCID being mice, they decided to use this humanized PBMC in this humanized mouse models. And what they did is they, for the control, they use the volunteer PBMC. It is the patients that does not have the diabetes and the patient that has diabetes, and they did the same experiment similar experiment that they did in this step 2. However in this case both the mice survived because the volunteer PBMC does not have volunteer PBMCs this volunteer PBMC cells has also already has this Volunteer PBMC's mice does not have this diabetic conditions, whereas patients PBMC mice has diabetic conditions. However, when they introduce this STZ induction, both the mice are now has this diabetic conditions, and both the mouse are the diabetic mouse. And when they introduce these E-isolate cells in uh, both the mouse through the kidney capsule transplantation, they can see that both the mice are survived. And when they check this same the glucose level, they can see the glucose level are more or less similar, whereas just this volunteer PBMC has high glucose level. Since it does not have one of the reasons could be that they does not have any diabetic disease, so their glucose levels are already at a slightly higher level compared to the patients who are affected with this type of diabetes. And when they check the human C peptides, they can see the human C peptides in both the conditions, that is patients, humanized mice, and the in the volunteer mice they do not these human C peptides are undetectable, both in the case of this first week post translation and second week post translation they do not see any humanized C peptides in this volunteer humanized mice model, whereas for the patient mice model they can see the human C peptide conditions. So the reasons could be that the reason could be that the in the volunteer humanized mice models, since there is no diabetic conditions, there are no insulin spike occurs. There are no insulin spike occurs in the case of this volunteer humanized mice model. Now, when they did this experiment, so they also when they did this clinical trial on the patients, they check different parameters such as insulin dosage, and they can see that after a certain time, it is in terms of this x-axis represents the number of weeks. So after few weeks, the insulin dosage are reduced, and also this TIR and TITR are also uh, been regulated within a range that can be seen here. That is insulin dosage that are measured by this TITR, insulin use, and HbA1c. These hemoglobin contents are HbA1c contents are continuously decreasing, and the insulin use is also reduced. And also this TIR and TITR levels are also within range. So, and apart from this, they also check this blood glucose fluctuations. So before treatment, they can see that glucose is fluctuate. Sorry, yeah. Before treatment, they can see that glucose is fluctuate too much, whereas after treatment, they can see that glucose levels are in line, are almost at the same average level. And the other parameters such as C peptides, insulin levels, and glucose levels are also measured, and they can see that pre-surgery. Uh, these are the pre-surgery levels of the glucose that are indicated by the dashed line, and the post-surgery, that is after 105 week, they can see that there is a decline in the glucose level, and also these insulin levels are also insulin levels are increased compared to these pre-surgery conditions, and the C-peptides levels are also increased compared to the pre-surgery conditions after 105 week. So this data indicates that this possible treatment, this treatment could be possible, uh, in, or this approach could be possible for this in order to treat the type 2 diabetic patients. So I hope you got a basic idea what is this type 2, how these type 2 diabetic patients are being treated recently. And if you need to understand more about their treatment process in detail and need to check their data, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.